Hello, everyone. My name is Nick Hall, and I'm an undergraduate researcher in Dr. Stephen Kolb's lab where we study neuromuscular disorders, predominantly ALS, amyotrophic lateral sclerosis, and SMA, spinal muscular atrophy. Today, I'm going to be talking about a project I've been working on to quantify RNA expression in our ALS-associated mutant mouse model. So what is ALS? ALS is a neurodegenerative disease defined predominantly by motor neuron death. Patients experience progressive muscle weakness that typically results in paralysis and eventually death within 5 years. Kinesin family member 5A, or Kif5a, is a gene that has been linked to the development of ALS. It is a neuronally expressed gene that encodes a protein for the kinesin motor protein family, which means that it is responsible for transporting cellular cargoes. Specifically, Kif5a moves down the axon of a neuron along microtubules in the anterograde direction, meaning from the cell body to the end of the axon. This is important for many different reasons, since molecules and organelles must constantly be moved along the length of neurons in order for them to function properly. To study Kif5a in vitro and in vivo, we created a knock-in Kif5a mutant mouse model with a guanine to adenosine mutation in the C-terminal cargo binding domain. As you can see here in figure 1, the mutation is located in intron 27 at the 3' splice junction of exon 27. This is because it is believed to disrupt normal splicing and cause skipping of exon 27, thus creating a mutant RNA and protein. We expanded our mouse colony to obtain wild-type mice with 2 wild-type alleles, heterozygous mice with 1 wild-type and 1 mutant allele, and homozygous mice with 2 mutant alleles. We then performed experiments to quantify RNA expression for each genotype. To do this, we designed an assay to measure the wild-type and mutant mRNA isoforms. We used 15-week-old and 30-week-old mice with n equals 3 for all groups. We first dissected the mice and collected spinal cord tissues. We homogenized the tissues using beads and then isolated the RNA through a standard phenylchloroform extraction. The samples were then treated with DNase in order to remove any remaining DNA and obtain as pure of RNA as possible. Reverse transcription PCR was then performed using purified RNA to create first-strand complementary DNA, or cDNA. This cDNA can then be used as a template to produce messenger RNA, or mRNA, through a technique called droplet digital PCR, or ddPCR. Since RNA is highly unstable, it is important to perform our experiments in this way and quantify the RNA as it is being made. ddPCR is similar to normal polymerase chain reaction, but prior to amplification, the sample is first suspended in small beads of oil to generate tens of thousands of these small droplets, which are then measured independently. Thus, this technique is highly sensitive. Our 2 assays detect the different forms of mRNA being produced, wild type and mutant. Both assays use the same primer sets, but have different fluorescent probes. As you can see in figure 2, the forward primer is located within exon 26, and the reverse primer is located within exon 28. The wild type probe is only able to bind exon 27 of Kif5a. Mutant Kif5a does not have this exon, so the wild type probe cannot bind to it, making it specific to the wild type mRNA. The mutant probe binds to the junction between exons 26 and 28, which is only present when exon 27 is deleted in mutant mRNAs. The primers and probes bind to the cDNA, and during amplification, as the polymerases move along the template strand, the probe is kicked off and emits a specific wavelength of light that is measured by a machine. We can then quantify the number of positive transcripts that contain the probe and negative transcripts that did not. We also quantified mRNA production of another gene, YWHAZ, which is expressed in all cell types and thus is a good normalizer for gene expression. Figure 3 shows our results from these experiments. As you can see in panel A, our 15-week heterozygous mice had about half the amount of wild-type Kif5a when compared to wild-type mice of the same age. Homozygous mice had near 0 amounts of wild-type mRNA. When we look at the 30-week cohort, it is not as clear of a story, with the heterozygous mice showing a 14% decrease in wild-type Kif5a. Panel B shows the mutant Kif5a expression. Heterozygous mice had a 40% increase in mutant Kif5a, and homozygous mice had an 80% increase when compared to wild-type mice at 15 weeks. However, 30-week-old mutant mice had much lower expression of mutant Kif5a than 15-week-old mutant mice. The 15-week wild-type mice had near 0 amounts of mutant Kif5a mRNA, as to be expected. Panel C shows the percent inclusion of exon 27, calculated as wild-type Kif5a divided by total Kif5a. Panel D shows and reflects total Kif5a expression with the wild type isoform in blue and the mutant isoform in red. Panel E displays the same information as in D, adjusted to a total of 100% to directly compare the expression ratios between the groups. Our results confirmed the expected levels of Kif5a mRNA at 15 weeks in a conventional knock-in mouse model of mutant Kif5a. The 30-week cohort did not follow the expected trend, which could be due to multiple different things, including possibly nonsense-mediated decay or age-related compensation. We plan to do future studies in order to understand why this is happening. We will expand our studies to include larger cohort sizes across different age points, as well as comparing different tissues. Well, I thank you for your time, and it was a pleasure to be able to talk to you about some of our most recent findings.